Hi everyone, today we will be talking about gene regulation. We will use the lac operon and the trip operon model to explain negative and positive gene regulations. Before we get into the details, let's first go over the basics. To understand how the operon works, we must understand the components of each part. Let's use the lac operon model as an example. The lac operon consists of an operator, a promoter, and together the gene they control. The operator is an on and off switch for a certain segment of enzyme coding genes, while the promoter is a site where RNA polymerase can bind to the DNA and begin transcription. All the genes are coordinately controlled. Now let's look at how the operon is controlled. The lac operon is named after lactose. It is an inducible operon, meaning that it is usually off but can be stimulated to be on when a specific small molecule, in this case an inducer, interacts with a different regulatory protein, in this case the repressor. The lac operon is innately active, and in the absence of lactose, it switches off the operon by binding to the operator of the operon, thereby blocking RNA polymerase from making the enzyme to break down lactose. To make the operon active, a specific small molecule called an inducer inactivates the repressor and turns on the operon. In the lac operon, the inducer is allolactose, an isomer of lactose formed in small amounts from lactose that enters the cell. With the presence of lactose, and therefore allolactose, they bind to the lac repressor and alters the shape so the repressor can no longer be bound to the operator. Without the repressor, the lac operon can transcribe into mRNA and make the enzymes that break down lactose.
The trip operon is composed of 5 genes. These genes are coordinately controlled, which means that it is controlled by a single on-off switch, the operator. The trip operon is a repressible operon because transcription is usually on, but it can be repressed with a repressor. The trip repressor, which is encoded by a regulatory gene, TRPR, is an allosteric protein, which means that it must be activated before it can serve its function. Tryptophan acts as its co-repressor when it binds to the repressor. Thus, the activated trip repressor binds to the operator and blocks the attachment of RNA polymerase to the promoter. As a result, transcription cannot occur. This creates a cycle maintained at equilibrium. The production of tryptophan is inhibited only when tryptophan accumulates in the cell.
On the other hand, positive regulation relies on E. coli bacteria's ability to sense the glucose concentration and relay the information to the lac operon, thus regulating the gene. Positive regulation can be explained in 2 scenarios: when glucose concentration is present and when it's scarce. This interaction depends on an allosteric regulatory protein known as cyclid AMP, or simply CAMP. CAMP accumulates when glucose is scarce. When glucose is present, the activator, CAP, remains inactivated and transcription is at its normal rate. However, when glucose concentration is scarce and lactose is present, CAMP concentration rises and CAMP binds to CAP and activate it. Activated CAP assumes active shape and can attach to the promoter of the lac operon and increase the affinity of RNA polymerase, and in turn, boosting the rate of transcription of lac genes. To sum it up, if the amount of glucose increases, CAMP decreases, and CRP detaches from the lac operon, and RNA polymerase binds less efficiently to the promoter, resulting in a low rate of transcription. And if glucose is scarce, CAMP will therefore increase, and the CAP will attach to the lac operon and stimulate transcription, therefore why positively regulating the operon to make it more efficient. The lac operon is actually under both negative and positive control. The lac repressor negatively determines if transcription occurs, and the CRP positively determines the speed of transcription.